All right. So how do you want to do this? How do you want to arrange the discussion?
I think we could start by trying to. I don't know a lot about Michael, so Homer, and I would like to know more about that.
Introductions. Introductions.
Yeah, I think I could try to explain. Yeah, a brief description of I think would be. So, Khalil knows me and Joe. My name is Isaac, and I'm the software member of the software team. And we try to help Friendzymes in general to create software that be helpful in the wetware and hardware to build this bioscience engine. Khalil?
Yeah. You were breaking off. So you were doing the introduction, right?
Yeah.
Okay. Cool.
Oh, another thing is, could you explain more about this? What do you know about software? Like, what's your experience with software? Because I think you try to understand better what would be interesting to explain and what things are redundant to explain.
Isaac, I think I cannot hear you.
Oh, very clearly. Hello. Are you hearing me better right now?
Yeah, better.
Yeah, that's much better.
Okay. So, if you could introduce yourself and explain a little about your software experience, could be useful for trying to understand what things we could explain and what things would be redundant to explain. I think would be.
Mitch, it might be you might be the best one to explain that. It's like, what do you want to know about software in synthetic biology?
Yeah, I just started my reading first at the iGEM blog. It's a very interactive resource, and I find it really easy to follow. Um, my interest really in synthetic biology is centered on primer synthesis. So I have this vision that we get to manufacture our own primers, because right now in the Philippines, all primers are sourced internationally. So we have two sources. First one is in Macrogen, Korea, and the second one is IDT in Singapore. And it usually takes around 2 weeks before before the primers arrive to us. That's after like the payment has been confirmed. So it takes quite a while to buy primers, and sometimes when we are doing optimization works, especially for just basic PCR detection assays, especially for viruses and some quite difficult to amplify targets, we usually need to redo our primer design somewhere in the middle, and add some degenerate bases, or use a different reference because something is just not working. So it takes quite a while to optimize. From my previous experience, it took us 2 years to 2 years to optimize genotyping protocol for rotaviruses, and by the time we were able to optimize the detection assay, our samples were already non-viable. So it was such a waste. It took us 1 year to collect those samples. So basically, first is on how do we the paper that I think it was who was it that sent me the paper in Twitter, Khalil, wherein they have a protocol on how they can scale up production of oligos in like in a petri dish, or because from what I've heard, it's usually a solid phase synthesis, but then they I think it was also
Isaac Larkin.
Yeah. Oh, yeah. So basically, I'd like to learn on software design on how to do that, because I cannot picture yet how you apply your software designs in the lab. Like if you have all those designs, like the plasmids and where the inserts are being placed, like how do you scale that up in the wet lab? I cannot imagine that yet. That's also I'm also interested to learn about that, because we currently started exploring using AmpliSeq panels. And AmpliSeq panels right now are very expensive. Like it costs $500,000 for just a 6 reference genome, for a whole genome, for One Health genomic surveillance, specifically for zoonotic viruses. So those are some of the applications that I'd like to use that synthetic biology technology. I hope that that's clear. Thank you so much.
Yeah, I very clarifying about what troubles you have right now and what they could provide to try to help you with it.
Also, when perhaps when Almir introduces himself, maybe you could also explain what you want to learn about nanopore sequencing.
So
Yeah.
hi, my name is Salmiro. I'm from Brazil too, like Isaac, and I'm part of the Friendzymes team and helping, a lot of ways. So yeah, so I think that our team wants to do nanopore sequence for confirming our constructs. Like we need to know that everything is all right in the right place. So we kind of use mini ion that's frugal. That's the most frugal sequencer to use. So it's a very good equipment to have. So we want to use it to confirm our plasmids and to see if everything is all right. Sometime in the future when we do the constructs.
A lot of times will be something like, now we have something that works,
Yeah.
but in laboratory, so we have some organism that is producing a lot of enzymes, but we don't know why exactly. So we have the phenotype that it's an enzyme that's producing a lot of enzymes, but I think that it's because of my design. So I want to sequence to have the genotype, the exactly gene sequence that is inside the bacteria to confirm that my design is what actually is making this phenotype. So kind of the confirmation process, like, or could be also like, I'm trying to make this work, but it's not working at all. Let me try to sequence this and see what's happening.
Yeah.
Okay, this is another way to think about it. But in general, we'll be sequencing plasmids. You know what plasmid is, right? Perfect. And also in the future, we'll be sequence E. coli or sublase or Pichia using the medium, because in the future we'll be integrating our plasmid in the genome to be more secure and resilient to genes. Okay, so I think that's it's our practical perspective. The software perspective, I have some knowledge about the problem of align the reads through some genome that's already sequenced and use this alignment to create the final product that will be the sequence that we sequence. So yeah, basically that's what I know for software. I know some softwares that do alignment. I understand the process that I receive a read, I align this, and I have a quality control process between reading and alignment, and I made the alignment and I get the final result. I kind of understand the pipeline, but I never made a sequence by hand. Like I don't know the whole process of sequence. So I don't know which specific things are related also to the minion sequencer. So I don't know a lot about this, and will be also interesting to know more about this specifics of using minion for this.
Cool. Homer, anything to add before we start the conversation?
Come in, Khalil.
Anything to add,
Say
or
again.
and what? Maybe you could introduce yourself, and maybe you could add anything you wanna include
Oh,
in the
yeah.
agenda.
Okay, so hello all. My name is Homer, and um, I'm here because I want to complete my the full bio stack. So you have read, you have write, and you have storage. So I want to complete all of those levels from a software developer's perspective. So I am an IT. My training is in IT. So majority of my skill sets are are behind the computer. So in silico. So I joined this project to learn a little bit more about the wet lab aspects as well as the design aspects of the what do you call this of the process. So I joined Khalil and Mitch with a previous project using the Nanopore sequence. So more or less, I kind of have a hand of how the read works. So now it's time for me to learn how to write the sequence that I want to write. So that's about it.
Cool. So maybe we could start with the software explanation, and then Mitch could go into the Nanopore explanation. Is that you okay with the plan? Cool, cool. All right.
So let's go. I think I will first start showing. So I think the first step in this journey will be try to explain the poly package. So poly is a project from Timothy Styles. Tim is now a friend. He created this package in Golang for engineering orgs. So this project started with Tim and Keone. Keone is one of our advisors to the Friendzymes project. And basically what they are trying to do is create a foundation where you could apply this package to use genomic for synthetic biology, basically. Okay, so I think we could start by understanding how we can use poly with for resolving some problems. I think would be a good use. So I created the Friendzymes toolkit. This is a repository that uses poly underline as a basics. And so okay. So one of the problems that Isaac Larkin shared with me is that they have some enzyme and they want to transform this enzyme to a sequence that could be synthesized and could be used by a host genome. And one of the things that we know right now is that each, instead of bytes, organisms use codons to produce amino acids. So we have 20 amino acids, but nature creates a way to have preference for some codons that is translated, compiled to each amino acid. Okay, so first, that's what I'm explaining. It's okay, or I have to step back to try to explain codon and amino acids.
Sorry, can you repeat that, Isaac?
Everyone knows codons and amino acids, Isaac.
Perfect.
Okay.
Thank you.
Yeah.
So, so the idea here is that we have to try some strategies to codon optimize some. Okay. So how codon optimize actually works. So you take a genome or like a human genome, and you take all the coding regions of a human genome. And now we have all the codons that they use to translate to some amino acid. And you count each amino acid for each, the counting of each amino acid to some codon. And now we have like a frequency of this. And you could use this frequency to say, for example, okay, if I want to make a project where my gene will be expressed in a human cell, will be interesting to produce my coding region using this codon table that in the final result will be a codon that have kind of the same frequency in codons to each amino acid as we have in human genome. Okay, so perfect. So the first thing is I try to create each codon table for Bacillus subtilis and E. coli, because this is the two hosts that we'll be using. And to make this easy, I use the poly package. So the first thing is I create this list of FASTA files with codons, with coding regions. So this FASTA has a lot of sequences, and each sequence is a coding region, right? One for subtilis, another for E. coli, and we have other strains of subtilis here, right? So my idea here is the wetware team asked me if I could create codon tables for each one, and I'll use this loop to iterate in this list of FASTA files, and I use FASTA.read to read the file here, and now I have all the sequence already parsed to FASTA to a Golang structure. And I could use this. So now that I have this, I will create a big string with all the coding regions inside. And I will use codon table.optimize table. This is a function that we have in Poly to create this codon table with the coding regions of some genome, basically. So the process of creating this table is not so long. Don't take so much. It's not so difficult to understand the idea, the original idea of what is a codon table, and this is provided by the poly package. Because if I don't have the poly package, I couldn't make I couldn't optimize a table with the coding regions of some organisms, and I could not it would be much more difficult to read FASTA files in general. So with Poly, you can see that I import sub package of Poly for FASTA, for GeneBank, and other things here to use Poly in my software. You are hearing the sound of some trucks or not?
Sorry, can you repeat that again?
Yeah, I have a truck in my window, so I am asking if you are hearing a lot of noise or not.
Okay, the audio is not clear with me.
Okay.
Khalil, can you respond?
Yeah, I'm only asking if because a truck is passing through my window, if you are hearing the noise of the truck.
Yeah,
Yeah.
but it's okay for me.
Okay. So the poly package also have other features, like we have packets for make reverse complementary of sequence, the complement of sequence, the reverse of sequence, the complement the base pairs that complement some sequence. We have we could also create variants, like I will show. One thing that a lot of times you can make is here. I want to create all the variants of some primer. So let's say that I want to have this primer to. So I have 10 base pairs that I know that will be in my primer, but the last 5 base pairs I don't know. I want to know every variant of 5 base pairs in the final part of the primer. So we could use this all variants UPack that you pass a string, use the UPack model, the UPack symbols, and you could create all the possible variants using this. That's another. We have also some a primer package, and this primer package has also other features, like you could calculate a thermodynamic for some sequence. So we could calculate the temperature, melting temperature of sequence, and we also have this function where you could create barcodes for some DNA, basically. And you could also create these barcodes passing a sequence of primers that you don't want to have in your sequence. It's another thing that is made. And we now, this is also a feature that it's not included right now, but we are making this possible. That is a software where you have given a sequence that you want to sequence and a specific melting temperature, the software creates the parameters for you for the sequence. So it's another thing that we are creating right now for Poly. For import and export, we could import and export FASTA files, GenBank files, GFF files. We have this poly.json that is basically, you could take this formats that we have here, like FASTA and GenBank, and make a generalization to a JSON file, basically. And we have a Rebase. Rebase is a database that we use, and Uniprot. So you could import Uniprot sequence also. And let me see more things here. Okay, for cloning, we have some features that we use also, like we have a Golden Gate simulation, basically. So one feature that we have is that you could pass a group of sequence and the enzyme that you want, and they will try to simulate the experiment for you and check if everything will be okay in silico instead of in the lab. That's another thing that we are making. Yeah. So I use these tools that they have for making the software that Friendzymes use. And so with Friendzymes, right now, I'm working in some features that I will like to share with you briefly. So in the Friendzymes toolkit, we created one feature that we created is the finder. And the idea is that when Isaac or other member that is designing a DNA, they could use this finder to find problematic parts in the sequence that they are working on. So they could know if something's wrong and try to make a decision if they change or not this problematic part. Another thing that we make is I I I finished yesterday. I finished yesterday the communication with IDT API. So when you take a sequence and you want to synthesize the sequence using IDT, they have a software that checks the complexity to synthesize this part and show to you if this part is synthesizable or not. So they doesn't share the code, the open source code for this complexity score, but they have an API. So we use the API, we consume the API to check if the parts are okay. And we have right now. So another thing is that if you are working with IDT to make primers, right? So one of the things we could make is try to see if the API has some feature that could be useful for automatizing the process of creating primers. It's another thing that we try to do. So IDT could provide something that will be interesting to check and to automatize the process of creating the primers for sequence. Could be one way to take a look at this process. And I think we'll also create this Golden Gate. We use the Golden Gate simulation in the poly and made more specific, is more specific for the use that we have right now, I think. And in the final part, we have split set. And split set is it's a software that NAB have for fragment some sequence in parts that will be optimal. So we recreate, we read the papers that split set are uses as reference and try to replicate the same idea, basically. And I think right now is what we have in software side. I could try to explain more specifics about the Friendzymes project and how this is related to what we're doing, but I think we could stop right now and see what questions or what things we could do for be more specific about what we want exactly.
Yeah. Sorry. Yeah. Thank you, Isaac. That was a very interesting introduction. So I kind of get the functionalities of the softwares. I think the other example you gave, that was the same thing. The group of Mads Albertsen in Denmark were doing Khalil when they were modifying their Arctic primers to be able to increase the number of samples that they can barcode by. I think from 96 they can now barcode 4 by 96 per run. So that's also interesting. With that, if that technology becomes more available and many labs learn how to do that, that would save us a lot of cost because barcode kits are expensive, and also it dictates how many samples we can simultaneously run. So right now there's this perception that Nanopore surveillance is not that advantageous because you can only run a few samples. But if that barcode limitation has an open science solution, I think that would be really attractive for many labs to switch to nanopore sequencing for pathogen surveillance. Another thing is for G-block synthesis, we've recently contacted IDT for that. I think I'm not sure if this can be addressed by the software itself or if this is really the nature of G-blocks, because one limitation I found when we were using G-blocks is that if we use G-blocks for controls, especially if we're doing diagnostic assays for highly pathogenic organisms, wherein it's difficult to source a culture or any DNA or RNA material which can serve as a positive control. So we use synthetic controls instead. And for some cases, we might need to use controls, longer controls. Let's say for conventional PCR for gene amplification instead of shorter controls for a real-time PCR. But the challenge that we see right now is that uh, G-blocks at size more than 1 kb are not recommended for PCR applications. So I'm not sure how the software could perhaps address it. What else? I think so far those are my insights specifically for the software, but it's really interesting that we have these functionalities right now. Yeah, Khalil, how about on your side, Khalil or Homer? Do you have any insights?
My,
That you
well,
want
my
to
question is
share?
like, what are the proprietary standard solutions right now? It's like, what do you people use right now
Sorry, we
for
use for what?
for what Isaac? So Isaac's creating a open source software for
Yeah,
these
okay.
functionalities. What are the like current software being used right
Oh,
now?
I think for primer design, we're currently using a Beacon Designer. But we do that one by one. Like for every primer that we need to design, we have to design it separately. So we cannot do simultaneous primer design. And the other one for I'm not sure for the integrating barcodes in your primer, since we haven't done that before. But we're basically just using freewares for alignments, and based on alignments, you just tweak the sequences manually, and then we send the sequences to IDT. So that's just how simple right now we do things.
One thing that I think would be useful is um, programming this type of software is interesting for synthetic biology and any biology related problem, because as you could see, like design is it's a problem. It's a problem because if you take a lot of if you take some time to design, we're actually delaying all the other steps. So design is actually like a threshold for the how fast you could create something, right? Some
Yeah.
design. So software is good for resolving this because if you don't have a specific heuristic, you could try everything, basically. So imagine that we don't know exactly what is the perfect primer for your sequence. Program is very good because you could try all the primers possible in some range, in some specific range, and see what will be the best. And if this takes much longer than needs, you try to make some heuristics to not select some space of possible sequence in this type of problem. And I think for example, we could try to see how the design is made by a people and try to translate this for programming and try to make the same thing that they do actually. And this is almost what we're doing in Friendzymes. So for example, we are dealing with a problem that some parts that we have are not synthesizable at all. So But we have a problem that this sequence is a sequence that we don't design it. We are using something that already works. And I understand that the designing team is hesitant of changing something that already works. So one of what they share with me is the strategy that they use for making this synthesizable. So basically what they say to me is that they break the sequence in smaller parts, or they introduce some sequence that could be removed in the laboratory. So I try to understand how they create these sequences that will be inside the final sequence, or how they divide the final part in fragments. And I try to reproduce this in software in a way that they could create this automatically. And one thing that we could try to make is understand how the design process is working right now for primers, for some specific primers for specific tests, and try to understand how they choose this and use poly package and other algorithms that we use for trying to make this automatic. And also we could try to, okay, but IDT, we don't know if IDT will receive this part, right?
Yeah.
So one thing that we could try to make is see if IDT have an API that make this type of check. I think yes. And you could use this API to check if the sequence that the software find could be synthesizable. And if it's not, try another one and continue this process until we find a sequence that uh, resolve the problem and is synthesizable also.
I think for now, for the primer design part, most of us are doing trial and error. So the key bottleneck is first looking for the right reference sequences. So it's quite tedious to look for reference sequences. Sometimes we cannot see sequences in GenBank. Sometimes you have to go to other databases. So once we have sufficient reference sequence, then from there, the next bottleneck is just, well, we just have to do the alignment. And then depending on the application, the more challenging primers to develop right now are universal primers, specifically for pan genus detection of some viral families, because it's quite difficult um, to add degeneracies in the primer while ensuring that it's still we can be able to sequence that. Yeah, one limitation that we have right now is when we use the generate primers, we don't seem to be able to get a high PCR efficiency. So the product that we have is still very difficult to sequence, even for Sanger sequencing, because of the idea that the PCR efficiency is just low. Maybe that's also one thing that we can look into. Yeah, but what else? I think so far those are the few things that relates to me. Where is Homer? Do you have anything, Homer? Oh, Homer is not. Khalil?
No, I'm good. I'm good.
So my question right now is, what other things we could help? How other things we could help? Actually, I think I'm kind of trying to make an overview of what we made and some ideas, but I don't know exactly what we could help. If it's more about education, about how what we're doing right now and how Polypack works and everything, we could try to make this. If it's more related to some specific problem, we try to make a project and prototype this solution. So I think my next question is about how it could be more useful.
Yeah. Yeah. Sorry. Go ahead.
Yeah, exactly that.
Okay.
What they could do together.
So far, the way I understand it, I think the software part is I think quite solid. What I do not yet, what I cannot imagine yet is when you have those designs, how do you integrate that in the wet lab experiment? Like for example, if I have a plasmid design for like let's say multiple primers in a specific order, because maybe I would want to generate a amplisig or a panel of multiple primers. How do I apply that specific design to the plasmids that I'm growing in the lab? And then yeah, so that's something I cannot visualize right now.
You say a specific problem. It's about receiving the plasmids and what exactly?
Like the goal of the um like you have for instance you have a software, and then you have your sequence products, and I assume this design is like not the traditional way on how we like for instance we grow plasmids in the lab. So when you have when you have a very complex design, it just looks complex to me. How do you like apply that in the wet lab? Like yeah, maybe I don't think yeah, that's something that I cannot imagine.
Okay, so I think um, imagine that we sent you the final plasmid for sublist, and we ask Philippines to make a change. So we say, okay, so this plasmid is for uh, producing some enzyme, and I think will be very interesting if Philippines actually produce insulin for some reason. Okay, and we send the plasmid, and you receive the plasmid to have the open drone. So what I do next? So the first thing is that we receive a lyophilizated uh, plasmid in a lyophilizated way. And the first thing you have to resuspend the plasmid. So you will actually use a reagent to resuspend the sequence, and you could actually you make a dilution of the lyophilizates and insert reagents to make them possible to mix with others. Okay, so this part, okay, right now I have this plasmids diluted, some liquid, some reagent. So the next step is I have to try to transform this plasmid, or no, I will not make this transformation. I will use some enzyme restriction kit to break this plasmid in a linearized part. Okay, so we want to linearize your plasmid, and this means that right now the plasmid could receive other enzymes, basically.
Yes.
Okay, so we use like maybe BSA1 for this, I think, in the design. That we design using BSA1 to insert new enzymes. Okay, so and Philippines created this sequence that have the open insulin sequence, and they created also this. Let me close the window. Perfect. So how you check this? I think in the lab you have to run a gel where you could find delinearized plasma, right? Okay. So the next step will be taking your sequence, use the restriction enzyme to have the overhangs, and okay. So now you have uh, uh, a sample with the plasmids linearized, and you have other plasmid with the open insulin. And you take this and you put inside a plate, and you take this and put inside the OpenTron machine, and we run a protocol for making the the ligation of both samples. So the process will be, you have some reagents to make the ligation and everything.
The ligation process, does it ligate a single sequence at a time, or does it allow for the ligation of multiple sequences simultaneously?
You could make simultaneously, but the problem is that you only could do this inside one flask, one small flask. Because if you do all the mutations in only one flask, you don't know exactly what's ligated to what. So this could be a problem. So we could try to make each ligation. So let's say that Philippines is actually making ligation for 4 enzymes. So the final product will be the OpenTron using the plasmid, the linearized plasmid for each enzyme that will be a specific flask. The final result will be the ligation, the ligation occurring in each flask, in 4 flasks, one for each enzyme. Okay. So, okay, right now after the ligation, we run again the gel, and you could see that we now have a plasmid circularized, right? And the next step will be transform this in sublis or E. coli. So we will make the same thing. So, but right now we'll make a sample for the host genome, and you try to make a protocol inside the OpenTron to run the transformation. So we could use some protocols for transformation, like temperature transformation, or I'm trying to remember. Using electroporation for the transformation, you could try to use the samples, and so the final result will be a sample where we created this transformation, and the final step will be plating all with this sample that are already transformed.
Yeah, I think I kind of get the whole process. Yeah. My question earlier was a bit far-fetched because I was I was like focused on the paper that Isaac shared in Twitter. I thought that you're already in that stage. Okay, so we definitely can do that here. My question would be, what biosafety level is needed to do that? Or do we have to apply for permits for like, do you know, Khalil, if we need to apply for a regulatory permit to be able to do that?
What biosecurity you read right now? What level
We
of
have
biosecurity?
up to BSL2 only. But since
I
we are
think-
doing renovations, we might be able to have until BSL2 plus.
Yeah, I think we already have level security for transforming, making genomic transformations in general. So I think this is not a problem. Actually, if we're making PCR, you actually already have enough biosecurity for this, I think. I could double check with Isaac, but I'm kind of sure that we don't need more than this, actually.
Yeah, the level of biosecurity for doing this is level 1 because E. coli and B. subtilis is all non-pathogenic. So the problem is the organisms that you use, not the process. Like the process is not a problem, it's not given danger, but the organisms that you use to modify is the problem. So if you use E. coli and bacillus and other level 1
Yeah.
organisms, you don't have any problem.
Yeah, so I think we can definitely do that here.
Yeah.
Yeah. But
Is
it'd
that
be
the point?
interesting to look at to see how the platform can be scaled up, like for like up to the level of 100 ligations simultaneously. I'm not sure if that's possible, but that would be nice.
I'd say something. Okay. OpenTron, it's okay. I will make this for another perspective. You know Ginkgo by Works, the company.
No, I'm not familiar.
Ginkgo is the greatest biosynthetic company in the world. The greatest in the sense that they are the biggest one. Okay, not the best one, but are the biggest one. They actually are evaluated by $16 billion this year. And I think this is one of the biggest evaluations at all in any bio-related company in the world already. And they sell the idea that they have this all things are very high throughput, like they could produce a lot of. But one thing that Keone and I share is that they make this because they have OpenTrons pipetting systems, but a lot of times they have to use humans to transfer things between machines, actually. They do a lot of work. Yeah, they do a lot of work. They could produce a lot of, like, thousands of ligations at the same time. But to make this, they have to have like dozens of humans also. Machines and humans are not like right now in with the solutions that we have right now for pipetting, transformation, they're trying to automatize this thing and not really automobile complete. So you have to have this support for humans to make this possible. So if you have one machine, I think ideally you want one human with the machine to see if everything's working and changing something necessary. And so 1,000 ligations are seeing that like one ligation, it's one sample. So this will take some time for automatize, or you have to need a lot of machines and people to work in this machine also. So it's not so high throughput as they think they made us think, right? So for now, only to share one information. I'm trying to work right now with the protocols that OpenTron will run. So I'm trying to create this standardized protocols for OpenTron. And I'm talking with Keone. Keone is contracted by OpenTron to make these protocols for them, actually. And what he said to me is that I have to run a lot of experiments to troubleshoot to see if the experiment actually works fine. So one of the things that you have, so when you receive the OpenTron, we will actually have to run some experiments to double check if everything's working as we are trying to make. So we will actually have to work together to make this automation part work fine. Actually, it's not something that are already solvable. Oh, solvable. We don't have a solution right now for this. We have to work together to try to solve this. And yeah, this answer your question?
Yeah. So here at PGC we're just we're a small team. Right now we have 5 staff working in the lab, and we're quite loaded with a lot of responsibilities, especially in the middle of the pandemic. But when the biosurveillance maybe becomes established, then we might have a lot of time to be able to resume our research works. Although right now, most of our focus is really on COVID, but we really but our mandate as an institution is really beyond just COVID. We're also into science advocacy and capacity building efforts, especially to bring in these technologies to other laboratories within the region, not just within the region, but neighboring regions as well, especially those from Mindanao. So it would really be interesting to be able to pilot that platform, and then we'll have something to share or something that we can also teach the members of our consortium. So we have a consortium, a genomics consortium, but the activities of the consortium is not just limited to genomics. We're also doing many basic molecular biology and other mobile applications. So when we expand our center, we do have. I'm aware that there's a planned genetic engineering lab, so that would be in the near future. I'm not sure when that would happen, but there's a planned genetic engineering lab together with PGC Mindanao. So yeah, this is very interesting. It will open many doors for us, and it will also allow us to contribute to this open science initiative.
Guys, just time check. Perhaps it's good to move to the Nanopore conversation, and we could continue discussing synthetic biology software in the Discord. And also, Mitch, maybe since you were curious about how to apply that paper that Isaac sent, maybe you could just post it in the Discord. Maybe
I actually doesn't know about this specific paper, but we could try to take a look together and see what we could do to make this possible. Also,
Yeah.
I think
So
that's the
yeah,
idea, right?
because that's just one of our vision. Like, really would want to have our own maybe a mini primer factory, just enough to cater to the needs of the region. Because right now everything is sourced outside the country. It's expensive, it's time consuming, and it limits what we can do. Yeah. So maybe moving on to Nanopore. So
sorry. I'm so curious about that. This is one. Just ask Isaac. Isn't that one of the objectives of Friendzymes to be actually to have the biofoundry to synthesize your own primers? I mean,
oligos.
like you could,
Yeah.
it's general enzymes, but could you are primers like a possible product of that frugal biofoundry that we're building?
Yeah, let me think. So we are trying to produce enzymes, and enzymes are produced um, by organisms in general. So people that companies that produce DNA like IDT, they do work with organisms to make this actually. So we could try to understand how we could make primers using organisms for this. But the problem is this will not be so customizable because after you create some, like I remember that Isaac was saying about one idea that is having a plasmid with of primers, right? So we have this plasmid with a lot of primers and use one process to break all the
Yes.
plasmid, right?
Yeah.
The problem is for me is that how I could ensure that if I don't want one of the plasmids in this in this one of the primers of this plasmid, so how I could ensure that I only get one plasmid of all this plasmid. So
Oh.
yeah, so
Yeah.
how I could do this. But imagine that okay, let's think about. Imagine that the Philippines only needs they only need the plasmid that we already have in the all the primates that we already have in this plasmid. So it's only, the idea is only to break this with an enzyme and everything will be running. This is one possibility. But to design primers, to make something like this, like I want an organism to produce this primer. This is not a simple thing to make in organism. This is very difficult. And companies that make DNA don't make this using organisms right now. I know that WIS is trying to make this using enzymes. This is something that we are trying to make because we'll be much more cheap to produce DNA. But right now, we use chemical synthesis for creating these DNAs that IDT and twist make, not the biological way of doing DNA.
Yeah. So if it's the enzymes, that would still be very helpful to us, especially for the enzymes that we routinely use for our diagnostic applications, PCR applications, reverse transcriptase, polymerase. So that would still really be very helpful for us.
Yeah. But we could try to understand more about this problem and try to see if this could be created for using the bacillus or E. coli production, try to make this possible. But right now, I don't know how we could customizable create some primer using organisms without having to design these primers already. You could actually use this for, like, if you have a set of primers that you use a lot of times, you could actually use the plasmid strategy to have an infinite resource of this plasmid, right? So you only have to input the plasmid inside the organism, and you have a lot of plasmids, and you filter these plasmids and and you have new primers. The set of new set of primers, right? So this could be useful, maybe, but I think we could try to discuss more about how we could use this. Yeah, one thing that will be really important is how we could use the BioStack, the OpenTron software, and the design team to make a solution that is more related to what Philippines need right now. I think this could be very useful in both sides. So what things you want to resolve and try to share this and see what you could make for resolving this problem. What do you think, Karu?
Khalil, what do you think?
Yeah, that's pretty interesting. I mean, even with as Mitch mentioned, if we could provide an alternative, open source, locally made, cheap, that doesn't have to go through customs, then that would be. But by the way, Mitch, what are you using right now? Where is it made? Is it local or do you have to import it as well?
Everything is imported. So we buy all our kits outside. I think there's only the only instance that there's a locally made kit was the COVID diagnostic kit from Manila Health Tech, but the reagents and the enzymes that they use, I'm aware, is still sourced internationally. So the raw materials are still produced outside.
If we could make it here, that would be insane.
Yeah. Yeah, definitely. I'm trying to also make the. I think my main goal here is trying to make cheap as possible, because if it's cheap enough, reagents will be free.
Yes.
So imagine a world where we don't have to import anything, right? So I think this is actually very interesting in my perspective. But if we don't get very cheap, the Khalil idea of creating a company or make a partnership with a company that could sell these more cheap than imported enzymes would be also.
Probes, yeah, we'd be very interested to have locally made probes because probes are very expensive.
So we could it's another thing that we could try to think about. For the nanopore sequencing, this other part of the project, I think my main goal here is trying to see the software that you created for pipeline the reads actually. So I don't know. I could talk. I think Isaac and Scott and Ahmed that are working in the laboratory knows the protocols for sequencing some sample, but they don't know how to actually make this software step, in silico step of how you receive the reads and how you produce a final sequence with this.
Okay. So for the nanopore sequencing, the principle of the nanopore sequencing is that it's it's a third generation sequencing. Unlike second generations, you do not need to really make a library in the sense that you do not need to fragment your libraries, but that still depends on your application. So you can just sequence the DNA in its native conformation. And the way it reads the DNA sequence, it is drags the DNA sequences inside a nanopore, and by means of electrical signals, the changes in the electrical signals as each base passes through the pore that corresponds to a particular sequence. So after that sequencing run, it generates a FAST5 file, and that FAST5 file is a non-human readable file. So it's like noisy signals, and those signals are base called to convert them into the FastQC file. The FastQC file is now the raw sequence data, which contains the base calls and the Fred scores or the quality scores of each base call. What's nice about Nanopore is that it's a long read sequencing, and I think the longest fragment up to date in record is 2 megabase. So it can sequence maybe the entire genome of a small genome of a bacteria continuously. And since you are interested with the structural arrangements of your inserts in a particular plasmid, then I think long read sequencing is very appropriate because well, if you have a single read that spans 1 megabase, then you have a reliable evidence that this is the exact arrangement of the DNA inserts within my plasmids. Unlike when you do short read sequencing that you intentionally fragment your DNA during the library preparation prior to sequencing to generate short reads, and you have to align them wards using some algorithms to generate your contig reads, then in those cases, the limitation of short read sequencing is that it's prone to error, especially specifically if you are assembling reads in highly repetitive regions. So that's the advantage of long read sequencing like nanopore. So specifically as well, if you're interested with structural variants like large insertions and deletions, then a long read nanopore sequencing would be better able to capture those phenomena. Yeah. I think so far that's it. But nanopore sequencing is very direct to the point. It doesn't need a highly advanced technical training, unlike the Illumina platforms, which are quite complicated. So that's also an advantage of nanopore sequencing. So we're still fairly new to nanopore sequencing, but so far we did not encounter any technical issues so far. That we
My other question will be how you received the FASTA 855, right, with the non-human readable file, and you make some transformations until you have the final sequence, right? And you have a pipeline for this. And my question is, it's a custom pipeline, or you use a software, a nanopore software for this? It's
a free algorithm. So there are de novo assemblers, publicly available de novo assemblers that can generate a whole genome sequence from your contigs, and I think yeah, that's fairly easy. That's fairly straightforward because these softwares are available publicly. I'm not sure of the copyright.
Yeah,
You
so
could send these links for me if you could. I think would be very useful for us to try to take a look in the software that we're using right now and try to understand if you need to customize something for sequencing for the pipeline of the plasmid
Yeah. So
or not.
I think I'll ask Paul for that. We have a resident bioinformatician who's in charge with the softwares that we are using, but we can definitely provide the information.
I could also try to talk with the wetware lab if they need some tutorial or some educational thing for working and make the preparations of the lab in the last part, and could also try to see if you could automatize this process in the lab using the open source also. I think this could be also other thing that you could use your expertise for trying to create this.
Yeah. We have a staff that also specialize in transformation and cloning. I don't have prior experience to wet lab for that, but of course, based on our experience, it's quite easy for us to adapt a particular wet lab workflow.
well, so for our SARS-CoV-2 sequencing, what base caller did we use?
I think it's Guppy.
It was Guppy.
Guppy. Yeah.
Okay.
There are some. Yeah. I'll double check, but I think it's Guppy.
So the Isaac and Amiro, are you familiar with the basic what you call this bioinformatics pipeline of nanopore sequencing?
Yeah.
Okay, cool.
I'll try to see if you could reuse a lot of software that we already selected. So I think the bioinformaticians trying to look for software that works with the COVID project, and we could try to see with them how we could customize this for our purpose instead of I trying to read all the papers for it.
Mm.
Omer, you have the list of softwares too, right? Like you were the one who gave it to Paul.
Yeah. From the top of my head, the base caller, which is the primary, base caller that transforms the electrical signals into bases. That's Guppy. Although again, there are many there are open source alternatives, but the standard and the what do you call this? The basic functionality is with Guppy, and then there is a then we then use Medaka. Medaka is a wait, top of my head. So Medaka is the one that does the assembly of does the job of assembling the fragments together, including giving out the scores and everything else. So basically, those are the two major softwares that are being used. And MinION has graciously packaged everything together in they have an automated workflow software such that all you have to do is to provide these are your reads, this is the reference, and then just goes and assembles everything together. So basically that's
think,
yeah.
Yeah, I think we'll be mostly using de novo assemblers because if
Oh yeah, definitely.
there's not much alterations in the plasmid, then we'll be using a reference base but if many alterations are to be done, then I think it's better to use de novo.
De novo.
Okay, I'll try to take a look in the novel.
Also, you might want to take a look at the bioinformatics pipeline of projects with the same kind of sequencing. For instance, like I don't know.
Yeah, I was trying to see this, and we have so sequencing is something interesting because everyone has the perfect tool, but I have to try to understand the perfect for what. So I have to read each paper to try to see.
Yeah.
Yeah.
what exactly they are creating this. And I'm spending some time with this, so my idea is trying to use the Philippines experience to try to resource this more easily for what happened.
I don't know. So maybe I don't know if
Yeah.
Homer or Mitch could pitch in
Yeah,
identifying a good pipeline for the project.
good. I think
Yeah.
we could try to make a document where we share the ideas together to try to sample every sequence.
But I think a
Offer-
nanopore sequencing is a good choice because there are new algorithms that are being developed right now that can directly detect modifications, epigenetic modifications, because it's sequencing the DNA or the RNA in its native form. So the electrical signal variation based on those epigenetic modifications are readily available even in old data sets. So that would be interesting.
Good. I don't know this. Thank you, Mike. Very useful. And I have 10 more minutes because I'll have to work. I have another work for instead I have Friendzymes at my actual work that pays for it. But my question is, what other things? I think I have in my mind that I'll try to read the paper to try to make the primer factory in the Philippines. So how feasible is this? What will be our pros and cons for this? So what we actually could do and what things we could do with the biostack pipeline? What other things we could make for Philippines?
Sorry, can you repeat that, Isaac? It was quite long.
Sorry. Yeah, I'll read the paper and we could try to make a document trying to see how we could make feasible the primary factory idea, right?
Yeah.
And the second thing is that you could share with me a document with some ideas of pipelines that use for the Friendzymes project for sequence plasmids. And my question is if you have other things that I don't remember that we could make, right? So that we discuss it, but I actually not take action for this.
Maybe the idea of integrating barcodes within your primers.
Okay.
I think that's also very interesting because that will open a lot of that will solve a lot of barriers in terms of multiplexing.
Okay.
Which could also save a lot of cost for small labs.
Okay. So Michael, I could contact you so we could try to elaborate more this idea and we could try to make a solution for this. Could be? Okay.
Yes, definitely. We can communicate through email. The Discord is just too fast. It's quite difficult to track the conversation, but yeah, I'm checking it once in a while, but yeah, it's too fast. So
Okay.
maybe we can have an email thread on this.
Okay, so share with me some a draft idea of how we could make what could be the input and the output of this idea, and I could try to work together in this email thread, so more details and more.
Sure. I can prepare the document. It'll take some time, but I can prepare the document. I'll send it.
Doesn't need to be so formal. Could be more like a thread of ideas. Okay.
Sure. Khalil, on your end, anything on your end?
I'm good. How about you other guys? Amiru and Homer.
I'm okay.
Cool. So let's continue discussing over email. So if Discord is too insane, so let's anyhow, so many interesting ideas now, and then yeah, looking forward to making this happen. All right.
I have one last thing. Is I'm discussing with Giovanna from the human practices, and we are talking that you, Khalil, have made a lot of discussions that are actually very related to human practice. So I think it will be very interesting to try to. We think that your ideas about not only how to use the biostack for the future. I think all the discussion that you propose about how will be the future with this technology, I think is very related to human practices.
Sure.
And it would be interesting if you could talk in another meeting about your ideas and see. I don't know if you know, but human practices are how this technology impact the world and my community, in the communities, and it's basically what you do, right? Basically, what I think you thought about those type of.
Yeah, man. We couldn't wait to
And I think it would be
apply
very.
this.
Yeah, and I think it would be interesting to have a talk with Giovanna after some time to show to you and see what you could implement or start some implementation for
Sure.
these ideas.
Sure.
And okay,
All right. Let's set up the meeting.
thank you guys.
All right.
Okay.
Have a great day. Thank you so
Perfect.
much, guys. Bye bye.
Thanks,
Thank you
guys.
so
Bye.
much. It was nice meeting you all.